Good afternoon. We are going to discuss a very interesting piece of the nervous system, which is the cerebellum, its structure, its function, and its dysfunction at the clinical level. This piece of nervous system is very unique in terms that one of the major components, which is the cerebellar cortex, is organized as a stack of chips. We will discuss this more extensively near the end of the subject. But in the meantime, know that at the last part of the last century, there was a book that became a classic, a book published that was entitled The Cerebellum as a Neuronal Machine, and was one of the first attempts at neural networks. It was written by the Nobel laureate Sir John Eccles, Masao Ito from Japan, and from Hungary, our dear friend and mentor in many ways, John Sentagutai, whose work we will discuss along the various subjects of this series. Anyway, let's start with the objectives. What do we need to come out with it? Essentially, is why the cerebellum controls ipsilateral muscles. You're all used to the problem that one side of the brain controls the opposite side of the body, and that's true for the motor system, as we may have discussed at some other time. But here the cerebellum is peculiar. It controls the ipsilateral muscles. Then the strength, that means the force, is preserved. The strength is preserved. All right? Although there are multiple signs of motor dysfunction, but strength is preserved all along. Now, we have to also study the cerebellum in terms of its organization that follows the principles of sagittal slabs. We'll know soon what it's all about. Then we will focus on how the cerebellum controls limb movements and how it controls trunk movements, and how the workings of the cerebellar cortex that were used, as I said before, as models for neural networks, is modulating the output of the whole machine. And finally, we may touch, if we have time, on the possible molecular basis for motor learning, because the cerebellum not only controls the motor system, but also has a role in learning motor skills and in storing them. Now, let's put the cerebellum in context within the motor system. So you have here the midline. We always start with the midline. And here you have the motor cortex that you well know, with the subdivisions into M1, premotor, and M2. You may remember that M1 has to do with fine voluntary movements, whereas PM has to do mostly with movements that are initiated by sensory stimuli, and M2 has to do with movements internally initiated without any sensory input. Now, you know that the motor cortex gives rise to the corticospinal system, that at the level of the lower medulla, it crosses to the opposite side. So obviously this motor cortex controls movements of the opposite side. Then you have now two systems that control the motor system. One is the basal ganglia system, and the other is the cerebellar control system. Now, the basal ganglia, both of these systems, basal ganglia and cerebellum, influence networks in the thalamus here, all right, which in turn will project its own result of processing to the motor cortex. But look at how it is laid out. The basal ganglia will project essentially to a thalamic network that is called VA, ventral anterior nucleus, all right? And this VA is going to project mostly to both PM and M2. On the other hand, the cerebellum, the output of the cerebellum, it is crossed. It crosses the midline and comes to a network of the thalamus that is the ventrolateral nucleus essentially, and this ventrolateral nucleus would project mostly to M1. So you have here the gadgetry so that the cerebellum, all right, exerts its influence to the opposite cortex that influences movement on the same side of that cerebellum. That means that problems with the cerebellum will result in problems with the ipsilateral muscles. All right. Now, on the other hand, problems with the basal ganglia system, as we may eventually discuss, they are influencing, again, through the motor cortex, the contralateral muscles. Now, what's the role of the cerebellum? Well, we already mentioned some in the objectives. First of all, it regulates movement coordination. Right? When you do any kind of movement, several nuclear groups have to be acting synergically and in any possible way to execute precisely the movement that you want to do. Right? But in addition to that, and that's the classical role of the cerebellum, in addition to that, more recently, it has been defined to a great extent the probable participation of the cerebellum in learning of skill movements. We'll touch on that at the end, most probably. Now, the cerebellum has been known for a long time, for, 200 years, some of its function, simply by looking at patients that show signs of dysfunction, and then the patient dies, and it so happened that had problems in the cerebellum. But the signs of dysfunction are very, very typical, very critical. One is what is called ataxia. Ataxia is the movement incoordination. If I want to get my hand here, I go straight like that, but if I have a cerebellar problem, I go all over the place before coming into my target. That's ataxia. Second, you have dysmetria. Dysmetria means that there is a problem with the measurement of the metrics of the movement itself, right? If I want to reach my hand this way on any object, if I have a cerebellar problem, I can overshoot it or I can undershoot it, and then I have to correct it, and so forth. That's dysmetria. My mouse is playing tricks. Now you have also the observation that when you reach with your arm that is a deficit because of cerebellar problems, when you reach for the object in the incoordinated fashion, when it's close to the object, it shakes and eventually grabs the object. So that last part is what is called intention tremor, which is different than the tremor of the Parkinson that is at rest. This is a tremor that occurs on action. Then you have a peculiar word here, dysdiadochokinesis. Comes from the Greek, whatever. The end result is that these patients have problems with rapidly alternating movements. For instance, if you ask the patient to tap, palm and dorsum of the hand, I can do it perfectly. A cerebellar patient will have problems. Will have problems in doing this also, right? So that's this dysdiadochokinesis. Then, when you test muscle tone, and remember that muscle tone, let's say in upper motor neuron disease, is essentially an increase of muscle tone. Everything is stiff, particularly anti-gravity muscles and so forth and so on. And in basal ganglia disease, this is rigidity. All the muscles are increased in muscle tone. In cerebellum, there is hypotonia. That means that the tone of the muscle is so decreased that you can move it around, and it moves like a pendulum without really any kind of resistance, which is characteristic of the normal tone of the muscle. Now, there is also what is called the lack of check. That is when you try to extend the arm against the patient, you ask the patient to don't let me pull, and then you suddenly release it. The patient lacks what is called the check. I'm checking, see, I do like that, and it stops. In the patient, it goes way back, and it may hit the patient himself. So you have to be careful on the examination. Then you have problems, the same as most of these problems that I discussed before are referring essentially to limb movements, arm movements, and so forth. But then you have the movements of the muscles of phonation that are also affected by cerebellar problem, giving rise to a problem in speech, which is called dysarthria. There is an influence of the cerebellum on eye movements, and that we will go through most probably in the oculomotor system lecture. At this point, you can just keep it as a reference. Now, another problem that occurs in eye movements is the deficit in smooth pursuit, which is the visual following of the target. We'll discuss this in oculomotor system anyway. So at this point, we defer it. And most important, remember always that the strength, the muscle power is preserved. You can ask the patient to grab your fingers and it won't let go. It's perfectly well preserved. All right, these are the general signs of dysfunction. Now I can show you some movie clips that how we test these things. For instance, here you have what is called the finger to nose test. This boy had a cerebellar problem, and see how it functions. See the dysmetria, the intention tremor, all the things that we were discussing before are apparent in this kind of examination, which is called the finger to nose test. Then you can do that for the lower limb by asking the patient to slide the heel on the shin. If you try to do it yourself, you see that you do it very precisely, all right? Whereas in the patient, let's see here.
Move your leg up like this. If you could just take this heel, start at your knee, go straight down and straight back up. Just with your heel.
See the difficulty in sliding smoothly on the shin.
Let's do this side. Move your leg.
Yeah, obviously in this patient the problem is bilateral. Has problem with both heel to shin test. Here you have an example of this dialocokinasis, which is the problem in executing rapidly alternating movements. See? There is some clumsiness that even more marked here. So she just cannot do it.
And just flex up the elbows. Just like this. There we go. And I want you to just take one wrist and just rotate it back and forth. Just one side.
See the incoordination of the movement,
The other side.
particularly on the left side. All right. And finally, I said that the cerebellum has to do also with truncal muscles that are important for maintaining posture, erect posture, and for walking. All right, and I'll show you an example of that here, of this same boy. See, he's trying to walk, heel to toe, in tandem, and has great difficulties. He loses balance, he falls, he's being held by the examiner. So these are essentially how you examine for cerebellar dysfunction. All right?
1 foot, right?
All right. What about mechanisms? How the cerebellum does it? Well, to control the musculature in the way that we described, the cerebellum has to get information on the conditions of the muscles at the moment that the movement has to be executed, all right? So this incoming information comes from the ipsilateral periphery about posture and muscle tone by spinal and brainstem roots, all right? There are sensory inputs coming into the spinal cord and into the brainstem and ascending on the same side without crossing and reaching the cerebellum and giving that information on the conditions of the muscles. But in addition to that, the cerebellum has to know what the cerebral cortex of the opposite side, remember now, are going to program the movement. So the cerebellum needs these two informations, one from the periphery, how the muscles are, the other from the central nervous system, what program is going to use to make the particular movement. Now, once the cerebellum gets that information and processes it, as we're going to see later on, all right, there are outgoing commands of the cerebellum. What the cerebellum with that information is going to do to influence, as we saw before, through the thalamic network, the motor cortex of the opposite side. Right? The contralateral motor cortex is one of the targets through the thalamus, as we said before, and that's the origin of the corticospinal tract that controls movement of the opposite side of the body. All right? But there are other target structures of the cerebellum. One of them is the red nucleus, that is the origin of rubrospinal tract, another descending pathway that influences movement eventually. And then there is an output of the cerebellum to both vestibular nuclei, or both sides. And these nuclei are, for instance, origin of the vestibulospinal tracts that are going to exert a very powerful excitatory action on anti-gravity muscles. All right, so that's the general layout of mechanisms. Now we go to the organization of the cerebellum. You have here a section of the cerebellum, more or less horizontal section through the cerebellum. Cerebellum is like a fist, right? The size of a fist that is sitting on the posterior fossa there, right? And has a cortex. As you can, in this section, you can see that has a cortex, all this gray matter, with a lot of sulci, a lot of enfoldings, cortex, and then has a white matter underneath, and embedded in the white matter has nuclei, the intrinsic nuclei of the cerebellum. This is, as you see, this is more or less at the pons level, and we're going to see the connections already with the brainstem. Now, if you take a little piece of that and you enlarge it, then you can see here. These nucleus that look like convoluted here, another nucleus here, another here, and here the cortex. All right. And when you do a section, a panoramic section of the region, you see very well that all this cerebellum here is dorsal to the brainstem. This is brainstem, as we were discussing before, right? This is at the level of between pons and medulla. You see some crossing fibers of the pons, and you see already the pyramid. So on the one hand is medulla, on the other hand is pons. So it's just at the limit. But you see overlying it, as you can see here, so this is brainstem, right? And overlying it is the cerebellum. And in the cerebellum you have cortex, much larger, of course, there's a sample of it, and this is to show particularly intrinsic nuclei. These nuclei embedded in the white matter. White matter is dark blue here, all right? And these nuclei are, then you have a cortex here, and then you have the intrinsic nuclei that are the midline one, which is shown very well here, is the fastigial nucleus, all right? And then you have the interposed nuclei here and here. And then more laterally you have this convoluted one that is the dentate nucleus. Right? So you have the cerebellum has a cortex, has a white matter underneath, embedded in the white matter, this fastigial, interposed, and dentate nuclei. All right. Now we take another aspect of the organization. Here you have a parasagittal cut, you're parallel to the midline. And what you see, I'm showing this to show this is cerebellum, obviously, all right? And this is brainstem, right? But you see how the cerebellum is connected to the brainstem through 3 pairs of thick fiber bundles. And these fiber bundles are called the cerebellar peduncles. So you have an inferior cerebellar peduncle, that is this one. All right? And this contains afferents to both the cortex and the intrinsic nuclei. And then you have the middle cerebellar peduncle, this one, all right, that contains also afferents to cortex and intrinsic nuclei. And finally, you have here the superior cerebellar peduncle that has the output. So you have here 3 pairs of peduncles from which you can see that there is a big disproportion in volume between the inferior plus the middle that are all receiving information, all right, and the superior that is putting out the information to target structures. So there is a tremendous convergence of information within that machine, right? So at least 3 to 1. So that's something to keep in mind also. Now, let's see how a general principle of organization. If you have midline here, right, and you have the cerebellar cortex on one side, all right, and you have the intrinsic nuclei, and then you have the target structures on the opposite side. Remember that the output was crossed. You see that the input to the cerebellar cortex, as a what is it here? As a rule, input to cortex and nuclei, cortex. That's what I'm trying to show here. The input to the cerebellum goes both to the cerebellar cortex and the intrinsic nuclei, to both. All right? On the other hand, the cerebellar cortex, whatever it does, it projects that processed information back to the intrinsic nuclei. That means that the intrinsic nuclei act here as a comparator between the information that comes straight from the periphery and the same information after being processed by the cortex. Finally, the intrinsic nuclei are going to project crossways to target structures, which target structure, the thalamus, the red nucleus, and so forth. All right. Now, another important feature also to keep in mind, there are so many things in this structure, is the organization in slabs or lobes. What happens is that all the sulci, the enfoldings of the cortex in the cerebellum, you can see it in the lab, they are all transverse, right? In the transverse plane, all of them, the enfoldings of the cerebellar cortex. And so there are some of these enfoldings that are very deep, and therefore 2 deep ones are like dividing the cerebellum into an anterior lobe, then a posterior lobe, and then another little one that is so-called the flocculonodular lobe. As we are going to see, this functionally, it does not make lots of sense. Let's take a look at this that I borrowed from Purvis. All right, the cerebellum is here, right, and this is anterior, all right, and this is posterior. How come? Well, because it's like enfolded, you see. And now if we stretch it like here, it's like that, and now we are opening it up, all right? From here goes to there, all right? And now this will be anterior and this will be posterior, and that's what is showing here in a stretched fashion, all right? And you can see here that you can imagine, or you can think in terms of so-called sagittal slabs that go parasagittally all the way through all the lobes. So you have a midline one that is called the vermis, that is this one, and that projects to the intrinsic nucleus that is just underneath, which is the fastigial, as we saw before. And then there is a region next to that, in a parallel fashion, that is called the paravermis, and that will project to the interposed nucleus. And finally, you have this very laterally placed portion that is called the hemisphere, all right, that is going to project to the dentate nucleus. Now, on the other hand, in the old times in which we were dividing the bilobes, you have here, let me see, an anterior lobe that is like that, see? is this is the big fissure. So this is the anterior lobe. But you can see that the anterior lobe is mostly vermis paravermis. It's very little of hemispheres. And similarly, the posterior lobe, that is all that from here to here, is mostly hemisphere. And then you have the remainder, which is the flocculonodular lobe. All right, from a functional point of view, the sagittal subdivision of the cerebellum makes more sense, as we are going to see eventually when we discuss function. All right? So essentially, there's this lobar subdivision into anterior, posterior, and flocculonodular does not follow function faithfully, whereas the slabs do follow functions faithfully. All right, now we go into the cerebellar control of limb movements. Remember that we'll do it separately for limb movements and for trunk or axial movements. Now in this case, we trace the midline and we put on the one side the cerebellar cortex, the cerebral cortex with the corticospinal crossing at the level of the lower medulla. And then you have on this opposite side is the cerebellum. And we are going to deal with the cerebellar cortex of paravermal and hemispherically slabs, not with the very vermis or midline structures. And we know that this cerebellar cortex, from the previous slide, is projecting to the dentate and interposed nuclei. Right. Now we have the following. Yes, that I'd like to illustrate each of these boxes with corresponding sections or slides. And here I showed you this already, that this section that shows here the cortex, and here you saw the dentate nucleus, and here the interposed nuclei. Very good. Now, cortex, the dentate, interposed. Fine. Now you have the input from the periphery that comes from the spinal cord and the brain stem, and it comes into the cerebellum through the inferior cerebellar peduncle. Right? In this kind of input, at the spinal cord level, comes from a tract that is called the dorsal spinocerebellar tract, which takes origin in the gray matter of the spinal cord and ascends on the same side to the cerebellum and reaches the cerebellum through the inferior cerebellar peduncle. All right. This input, as you can see, and this will be always the same, because it's a pattern that keeps on repeating all along the cerebellum, it terminates in the cerebellar cortex, but it also leaves collaterals in the intrinsic nuclei. And you know that the cerebellar cortex projects to the intrinsic nuclei. So this is actually comparing, as I said before, the same information coming from the periphery and the same information after being processed by the cortex. All right. At the spinal cord level, you have here a nucleus, that is the nucleus of column of Clark, that is actually the origin of the dorsal spinocerebellar tract that I mentioned before. At the brainstem level, you have reticular nuclei that also feed, and let me backtrack 1 second. This origin in the spinal cord deals with the lower limbs. The upper limbs don't find the information that comes into the cord. There is no nucleus column of Clark, so they ascend to the brainstem and they terminate in what is called the nucleus accessory cuneatus. All right, and now the output of accessory cuneatus joins the inferior cerebellar peduncle to come into the cerebellum, and that's the inferior cerebellar peduncle, the cut of it, the myelinated bundle. Now, in this way, the cerebellum is getting information from the periphery. Now it has to get information from the cerebral cortex, from the planning of the movement, right? And there is a good outflow of cortical fibers that descend in the midbrain, all right? And at the pons level, they are going to terminate in the pontine nuclei. Pontine nuclei are all these groups of cells that are within the base of the pons, right, where these fibers from the cortex terminate. And many things happen here in the process of information. And eventually, these pontine nuclei have outputs, and this output crosses the midline and forms what is called the middle cerebellar peduncle, which is this bridge of the pons, right? That are going to terminate where? The same. Going to terminate in the cortex and also in the intrinsic nuclei. All right. Here I show you a section in which shows the base of the pons, and all the light areas are pontine nuclei, and the output crosses the midline and makes this middle cerebellar peduncle coming into the cerebellum. Now, one more structure has to be considered in addition to the information that cerebellum gets from the periphery and from the cortex, and that's the inferior olivary nucleus. Right? This inferior olivary nucleus receives input from the same cerebral cortex, receives input from the contralateral spinal cord, and the output will cross the midline as the olivocerebellar fibers and join the inferior cerebellar peduncle, making it bigger and bigger. Right? And here I have a section of that in which you see here the inferior olivary complex that receives these inputs from cortex and spinal cord, and the output are the olivocerebellar fibers that cross the midline and take the route of the inferior cerebellar peduncle to come to the cerebellum. And you see how the inferior cerebellar peduncle that here was relatively small, here becomes very large through the addition of these olivocerebellar fibers. Now, that's the olive. That's this inferior cerebellar peduncle. Now comes the output of the cerebellum. And this output, as you can see, comes from essentially from the dentate and interposed nuclei, and from this superior cerebellar peduncle that was the small one that carried the efferents from the cerebellum. This superior cerebellar peduncle crosses in the midline and terminates both in the red nucleus and in the thalamus, in ventral lateral nucleus of the thalamus. I'll show you a section here. Yes, this section, this is caudal midbrain, and this is rostral pons. And you see this massive decussation here in the midline, all right? These are the two superior cerebellar peduncles crossing. And then you see that's the crossing there. And then in more rostral sections, you see the red nucleus here, and you see the thalamus here, right? And eventually, the output of the thalamus goes to the cerebral cortex, as we show here, and the output of the red nucleus now immediately crosses the midline and descends in the opposite side as the rubrospinal tract. So you have now two descending tracts, the corticospinal and the rubrospinal. But perhaps more important than that, particularly in human, is the fact that the red nucleus also projects an important projection to the inferior olivary nucleus. Now you see here that there is like a triangle. You know, it's not easy to see because these are all orthogonal lines. But if you take a look, from the dentate goes to red nucleus, from red nucleus to olive, from olive to cerebellar cortex, from cerebellar cortex to dentate. This is a famous classic triangle that I think I'm going to show you. I'm going to show it to you later because the effect of lesions there is characteristic. Wait a second. All right. So what happens with lesions? All right. You have lesions of the cerebellar system in any part of the system that will impinge on limb movements of the ipsilateral side of the cerebellum. So where can be the lesion? Well, the lesion can be even like that in the spinal cord, affecting the dorsal spinocerebellar tract and even the ventral spinocerebellar tract all these inputs from the spinal cord to the cerebellum are a deficit, and you will have a cerebellar dysfunction on that side. Or the lesion can be in the cerebellum itself and give you the same thing, problems with the cerebellar function on the ipsilateral side. But the lesion could be here, all right? This is the midbrain, affecting red nucleus, all right? And through the red nucleus, very close, is the third nerve, all right? And here you see that you will have something like a cross syndrome, because on this side you may have a third nerve, and on this side you have ataxia, because you are interfering with this system. So it will be a cross syndrome, so the lesion again will be in the brainstem. Where in the brainstem? In the midbrain tegmentum, right? So this is tricky, right? Because we are telling you that the cerebellum controls movements of the ipsilateral limbs, and that keeps on being true, with the little problem of a lesion here in the midbrain that could give rise to cross-cerebellar signs. It's a little tricky, but you have to think about it. All right, this is the triangle that I was referring to you before. See, from dentate, it goes to red nucleus, from red nucleus to inferior olivary complex, from inferior olivary complex back to cortex and dentate nucleus. So this has even an eponym, the triangle of Vienna-Moyaret. And the interesting thing is that when there are interference with this triangle, the patient may show a very peculiar symptom, which is palatomyoclonus. You know, the soft palate clicks all the time. The patient can hear the click, the examiner sometimes hears the click and never stops. All right? And that's one of the occurrences is when the patient has a tumor of the cerebellum and the surgeon resects it. All right? If it resects it deep enough to affect the dentate nucleus, that patient probably will have palatomyoclonus on that side. So that's a clinical parallel, let's say. All right. Now we go to the control of axial postural muscles. All right. Now this is a system that involves majorly the vestibular system. There is a lot of correlation between cerebellar and vestibular function. They are very tightly connected, interconnected, as we're going to see. In this case, we are dealing now with the sagittal slab, which is the vermis of the cerebellum, cerebellar vermis, right? Way in the midline, all through the lobes, right? And also the flocculonodular lobe. The underlying intrinsic nucleus is the fastigial nucleus, to which this cerebellar cortex projects. Right? Now, you have it here. You have here the fastigial nucleus, right? Fastigial nucleus. And here you have now the vestibular nuclear complex with the lateral vestibular and the medial vestibular. They are located at this junction of the brain stem here, where this is the location of the vestibular nuclear complex, of which two nuclei are important for this postural control, the lateral vestibular and the medial vestibular, mostly the lateral vestibular. All right? How is this working? Well, the vestibular nerve, both the nerve and the nuclei, project to the cerebellum, to the medial slabs of the cerebellum, again through the inferior cerebellar peduncle, and they terminate both in the cerebellar cortex and in the fastigial nucleus. The cerebellar cortex itself, the vermis, projects to the fastigial nucleus. All right. An important exception to the general rule of connectivity that I just mentioned all along is this direct connection of the cerebellar cortex now to the lateral vestibular nucleus. As we are going to see, the output of the cerebellar cortex is essentially inhibitory, and therefore this cerebellum is really applying the brakes on the lateral vestibular that is actually let me continue here in a moment. So we are talking about outputs of the cerebellum, a direct cerebellar cortex output to the lateral vestibular, and then you have the output of the fastigial nucleus that goes to all vestibular nuclei, and not only of the same side, but also to the ipsilateral, to the contralateral side. That's the fastigial. Now, the lateral vestibular is very important because it's the origin of the vestibulospinal tract, the lateral vestibulospinal tract that goes all along the spinal cord to actually lumbosacral levels, and it plays a role in the neuronal networks of the gray matter of the spinal cord, controlling the motor neurons. And this, the action of the lateral vestibulospinal is essentially excitatory of extensor reflexes. All right, and it's one of the main components of keeping you in an upright posture. Right, and now there is also an output to the spinal cord from the medial. These indicate. I put these to indicate that the lateral vestibular is going all the way to the sacral cord here. Right, but there is also an output from the medial vestibular nucleus that goes only to cervical levels, upper cervical levels. Right, they have to do with control of neck muscles. All right, you have to keep in mind also that the fastigial nucleus, the flocculonodular lobe, and the vermal cortex, they all also receive input from the inferior olivary nuclei, the same as in the previous panel that had to do with the control of the limbs, because the inferior olivary complex is exerting a very unique function for the entire cerebellum, for both all the slabs of the cerebellum, in terms of providing a timing impulse influence. So now we are going to discuss the actual mechanisms within the cerebellar cortex. For that, we have to know a little bit of its composition. And this section that has been treated with a GABA antibody, and therefore everything that is brownish or beige means that it has GABA and is of inhibitory nature. You can see here that this cortex consists essentially of three layers. First of all, you have a molecular layer, then you have a single cell layer that with the Purkinje cells that is called the ganglion cell layer, and finally a thick layer of granules that is called the granule cell layer. Now these granules are extremely tightly packed. It goes up to 2 million per cubic millimeter, and overall, if you add them all up, it has that layer of the cerebellar cortex has more neurons than the entire rest of the central nervous system. So anyway, you have here a list of the neuronal types within the cerebellar cortex. One is the granule cell, that is the input cell. It's excitatory and uses glutamate as a transmitter. Then you have the Purkinje cell, is the output cell, the axons of which will go through the granule cell layer and go into the white matter for the target structure. Is GABAergic and is inhibitory. Then you have within the molecular layer, you have other cells that are short axon cells. One that is indicated by that red arrow is the so-called outer stellate cell. It's inhibitory also. It uses GABA. Then you have also another kind of interneuron, which is the basket cell, that is closer to the Purkinje cell layer. We'll see how they connect eventually. Then within the granule cell layer, there are very giant cells of short axon that are called the Golgi cells. They are also GABAergic and inhibitory. So other things that you can see here is that the molecular layer contains all the dendritic trees of the Purkinje cells, of the granule cells, of, I beg your pardon, not of the granule cells, the Purkinje cells, the stellates, and the Golgi and the basket cells, all dendritic trees. And then within the granule cell layer, you see these spaces, these vacuum spaces that are lodging what are known as the cerebellar glomeruli that we'll see in a moment. All right. In addition to the neuronal types, you have fibers in the cerebellar cortex. First, all the input fibers terminate in the cerebellar cortex in two shapes, two tastes. One is a climbing fiber, which is a fiber that actually climbs the dendritic tree of the Purkinje cell. It's excitatory and comes from the inferior olive exclusively. And then you have the other pattern of termination is that of mossy fibers. They are coming from all over the other sources of input to the cerebellum, and they are also excitatory. So these are the afferent fibers to the cerebellar cortex, climbing fibers exclusively from the inferior olive, and mossy fibers from anywhere else. In addition to that, you have an intrinsic fiber, which is the so-called parallel fiber. Parallel fibers are actually the axons of granule cells. You see a granule cell in the granule cell layer will send the axon to the molecular layer. The axon will split into two in T shape, and there will be now a parallel fiber that goes through the entire molecular layer. Now, you can imagine that there are so many granules here that the molecular layer is pierced by bundles of axons of the granule cell, and because they are going in parallel, let's say, to each other, are called the parallel fibers. They are, like the granule, they are excitatory. Finally, the efferent fibers of the cerebellar cortex are just I mentioned that before, are just the axons of the Purkinje cell. They are strongly GABAergic, inhibitory on the target structures. What are the target structures? We mentioned that the cortex projects to the intrinsic nuclei, therefore intrinsic nuclei are a target structure. Also, we mentioned that is one exception to the rule that the cortex projects to intrinsic nucleus. In this exception, the cortex projects to the lateral vestibular nucleus, which is another target structure. All right, now let's see how they interconnect within the cerebellar cortex, and I'll do it in a very schematic way, all right? And it's extremely simplified. Here you have like a key of, of the abbreviations and all, but it's self-evident, as you'll see, and I make excitatory elements in red and inhibitory elements in blue. So first of all, you have the granule cell, right? It's the input cell, we said. And the granule cell is a small cell body and has local dendrites right there. Not only one, they have several, but they are just clustered, all right? These are the granule cell. Now, the granule cell is the input cell. The Purkinje cell is the output cell that projects to the intrinsic nucleus that is inhibitory. Has a huge dendritic tree that goes through the entire molecular layer, and actually it branches in a plane perpendicular to the long axis of the cerebellar folium. And in that way, in a rather narrow space, so that means that is actually what rules the organization in like chips, one parallel to the other, each one with a Purkinje cell branching as the main feature. Now, you have here the stellate cell, and the stellate cell is inhibitory, and as you see, it influences the dendritic tree of the Purkinje cell. Then you have the basket cell. The basket cell has a long axon that goes for quite a distance, and when it crosses a Purkinje cell, gives off a descending branch that is going to contact the axon initial segment of the Purkinje cell. Now you know, I'm sure, that the axon initial segment is where the nerve impulse is generated. And here you have an inhibition of that region. That means that the basket cell has the property of shutting off the Purkinje cell, because no matter what is happening to the Purkinje cell, it's inhibiting just the zone that will initiate the nerve impulse in the axon. All right, then you have the Golgi cell. The Golgi cell is a giant cell that is within the granule cell layer and has local dendrites and has a huge dendritic tree that goes into the molecular layer and branches in all dimensions of space. Just a second. That's the Golgi cell. Now we'll see this is the intrinsic connectivity within the cerebellar cortex. Now let's see what happens with the inputs. All right, you have here a mossy fiber input. Where is it coming from? From all over the place except the inferior olive. All right, inferior olive gives only climbing fibers. A mossy fiber that reaches the cerebellar cortex and leaves collaterals in the intrinsic nuclei. Remember that it's always that pattern, right? That ends both in the cortex and in the intrinsic nuclei. And you can see here that this mossy fiber is excitatory, all right? And is contacted here dendrites of the granule cells and of the Golgi cells and excites them. But here you have the gadgetry that the Golgi cell has a short axon that in turn inhibits the granule cell. So it gives rise to a complex synaptic arrangement here that is actually like enveloped by glia, and that's what's called the cerebellar glomerulus, and that's what the other slide showed as spaces within the granule cell layer. All right, so now let's put the climbing fiber. The climbing fiber, wait a second, no, excuse me. Let's put the axon of the granule cell. I said that the axon of the granule cell goes up to the molecular layer, splits in two, and it travels, forming what is called the parallel fiber system, and whenever touches dendrite of any other cell, it excites it. Now, finally, you have the climbing fiber. The climbing fiber comes exclusively from the inferior olive. It leaves collaterals in the intrinsic nuclei, the usual, and then climbs the dendritic tree of the Purkinje cell and blasts it with the law of excitation. So you have here a lot of gadgetry, all right, in terms of circuits. What is very noticeable is that there is a lot of inhibition within the cerebellar cortex. Look, all these blue elements, they are all inhibitory, right? The only element that is excitatory is the granule cell with the parallel fiber and the incoming mossy fibers and climbing fibers. All the rest is inhibition. So there is when a volley of impulses comes from the mossy fiber, let's say comes from the spinal cord, from the condition of the muscles or whatever, what's going to happen? It's going to, first of all, this arrangement of the glomeruli and the feedback from the Golgi neuron onto the granule is going to restrict the amount of granules that this mossy fiber is going to excite, because most of them will be inhibited backward by the Golgi neuron. So a small number of these axons are going to form the parallel fiber system. Now, the parallel fiber system eventually is going to discharge inhibition on the intrinsic nuclei. The problem is that you have all these other neurons that are also inhibiting the Purkinje cell. So you see that there is some kind of a balance between excitation on one side and inhibition for the other. And in the resting state, the balance favors inhibition. That means that in the resting state, this Purkinje neuron is relatively inhibited. And now it comes the climbing fiber, it reaches that dendritic tree of the Purkinje cell and samples the condition under which that Purkinje cell is, and blasts with excitation, and then the Purkinje cell will discharge according to its previous status of the internal circuitry, dictated by the internal circuitry of the cortex. I hope that is not too involved or not too difficult. All right. Now, if you have any questions, of course, what we are saying. As I said before, there's a lot of inhibition in the cerebellar cortex, and you can delineate circuits that use feed-forward inhibition and circuits that use feed-back inhibition. For instance, you have a feed-forward situation. You see, for instance, A excites B, B excites C, C inhibits D. This is feed-forward, right? And in examples, A can be a mossy fiber that excites a granule cell, which in turn excites a basket cell, which in turn inhibits the Purkinje cell. That's a feed-forward inhibition circuit. But you have the feedback inhibition, which apparently is very important, in which, let's say, a MOSI fiber will excite a granule, and the granule will excite a Golgi cell, and the Golgi cell will inhibit the granule. Right? So that's a feedback here. So the two types of inhibition are acting in the cortex. So as you can see, it's a very involved process. And the end result of the process is how much that Purkinje cell discharges and gives a certain amount of necessary inhibition to the intrinsic nuclei. All right. That's about that for the cerebellum as control of the motor system, coordination of movement and the like. Now let me finish with one very recent example showing the role of the cerebellum in motor learning now, not in the motor action, but in the learning of the skill. And for that, the paradigm that was used mostly is the associative eye blink conditioning. You know that you can touch or press the orbit on one side, press the orbit, the animal or the person will feel pain, all right? And in this case, shock, let's say. And if you pair that with a tone, eventually after so many pairings, the tone will elicit the blink to the stimulus, all right? So you have, therefore, something like that in the cerebellum. First of all, that the unconditioned stimulus, the shock, is encoded in the discharge of the climbing fiber, all right? On the other hand, the tone is encoded in the MOSI fiber parallel fiber system, essentially in the parallel fiber system, right? And the parallel fiber system is going to also stimulate here the Purkinje cell. And whatever, while it's not conditioned, each one does its own thing, and the Purkinje cell discharges adequately and inhibits sufficiently the intrinsic nuclei to provide for a very closely timed blink to the shock. Now, after pairing, what is happening? After pairing, repeated pairing, what is happening is the following, that the encoded tone now that goes through the parallel fiber system, at the level of the synapse with the Purkinje cells, there will be a process that is called long-term depression. Long-term depression as well as long-term potentiation, are have been considered cellular basis of memory, and in this case there is long-term depression here. So what is happening that since this synapse is depressed, when the climbing fiber doesn't have enough to stimulate the Purkinje cell, and the discharge of the Purkinje cell decreases. You see that it's thinner than here. It decreases. So the inhibition to the intrinsic nuclei decreases, and the intrinsic nuclei, which in turn are excitatory, produce eventually the timely blink. All this is fine and dandy, and has been shown electrophysiologically. Like the interesting thing is that now there is evidence that the long-term depression at this kind of synapse involves, first of all, the simultaneous activation of metabotropic glutamate receptors, which are at this synapse, all right, and that are activated by the parallel fiber system, simultaneously with voltage-gated calcium channels that occur here as a result of the discharge of the climbing fiber. All right? When the two things occur simultaneously, as it happens after repeated pairing, that results, this simultaneity of the two impulses results in an activation of a protein kinase C enzyme in the Purkinje cell, and the activation of the protein kinase is going to lead to phosphorylation of the glutamate receptor 2 that is here, is at the level between the climbing fiber and the Purkinje cell, and that phosphorylation makes that receptor to go inside the cell. So that means that is an internalization of the glutamate receptor. So this essentially, this Purkinje cell becomes less excitable, and that lasts a good period of time. All right, I hope that you can digest all this information, which was maybe too much, but take whatever you like from it. All right, and thank you very much, and we'll see you